Worst case scenario, the thing would explode, though I have trouble seeing that happen. So that's rinse number one. We don't got to empty our reservoir tubes because we just are rinsing and there's not enough volume because it hasn't gone through. 1, 2, 3, and I'm just going to double check something. Seconds. And so we're done with our washes. So now all we got to do is dry it out. That's going to be a 90-second spin. After we dry it out, we're going to elute into clean tubes with a little bit of water, and then we can proceed into our ligation. So about 8 more seconds. So when I peeked earlier, I noticed that the wash step didn't work that well. Again, I think it's some of that precipitate is still clogging up the zymo column, so hopefully a little bit more has gone through now. We'll soon find out. So here's our questionable tube. Not too hot. Well, we'll spin it a little bit longer and see what happens. So this will be our drying step for the 2 tubes that are working normally. And for the small fragment cleanup, this will be a step to try to force a little bit more of that wash buffer down. So we're going to spin that for 90 seconds. While that's spinning down, we're going to do a quick review of what happened. So we had some DNA. We digested our DNA. We then ran it on a gel. We took a picture of it, and then we cut out our little fragment on the gel. We then put it into some tubes, added some ADB. At that point, I should have been adding some isopropanol to the small fragment cleanup. I should have added 2 parts isopropanol to 1 part ADB, or about 400 microliters isopropanol, 200 microliters of ADB. I didn't, that's why I'm paying for it right now. And we put that at 55 degrees for about 10 to 15 minutes. That breaks down the gels, that melts the gels. After we get this liquid gel mixture, we can come and we can start to throw it on our zymo columns. And we just spin our liquid through the zymo columns, rinse it with PE, 250 microliters twice, and dry it. We're in the drying step right now. Again, you always want to be thinking ahead. You can see I already made the mistake here with the small fragment cleanup. It's very easy to make that mistake to kind of space. You want to be thinking ahead if you're doing small fragment cleanup. If you can avoid the gel purification, you want to, because you take a hit in efficiency there. If you just space and you do the gel purification, don't forget to add the isopropanol earlier. It may complicate things if you add it later. So here's the drying step almost done. Let's see if we manage to get our small fragment cleanup to work this time. One. Looks like it went through. So we're good to go there. And you can see that on the side of that tube we have all sorts of gunk. So that's probably why it didn't work too well. So I'm actually going to spin this for another 30 seconds just to get it extra dry. So the other 2 are probably dry right now, but our tube that is questionable, it's spun for 90 seconds in order to get all of that liquid out. I discarded the flow through, and I'm giving it another 30 seconds just to make sure it's extra dry. In the meantime, we can prepare for the elution. So we're going to be eluting in 15 microliters of water. So grab my pipette tips. Grab my P20, which is hiding from me. Oh, there we go. Thank you. So set my P20 to 15 microliters, and I'm going to grab my clean water, which is right here. So 15 microliters. And then I can grab my column, get rid of that flow through. We reuse those, and then place the column inside of a clean epi tube that has been labeled. Always be careful to double check the column and the tube that you're going into. This is a very easy place to make tube switches. And last sample. So V2 is going into V2. And then we just add in our water The zymo columns are pretty easy to add water to. It's a very small membrane, so it'll pretty much be forced to go into there. So you can reuse tips, and I'm just kind of gently putting some water right in the center there if I can. 1, 2, and 3. Cap everything, and then I'm going to load up my tubes into the centrifuge. Centrifuge has an arrow, points that way. That means this thing will be spinning this way, which means I want to have my caps facing the opposite direction, otherwise they may break off. And so I can just load it in and spin for 30 seconds. So cap, close, spin it. And at this point the water is going through, it's dissolving the DNA, it's going to flow through, and it's going to collect in our Eppendorf tube. And from there we're going to have DNA, which we can use to ligate things. So we'll see if it worked. Hopefully that small fragment isn't too clogged up and we'll be able to actually collect that water. Otherwise I get to redo some stuff. So cross your fingers for me. And at this point you can throw away the tubes that had your melted stuff in there. That's just junk, so that's gone. So here's our tube in question, and you can see at the bottom, sure enough, we have a little bit of liquid. That should have our small fragment, and we'll find out when we do our ligation if it works. So you can just take out your tubes, throw away your columns. You should probably be double checking to make sure that everything is labeled right. So V1, V1, and here's your 3 samples, each of them with their DNA, and you're good to go for ligation. Yay.